So we have seen how transcription activators can bind DNA, recruit chromatin remodeling complex, get rid of the histones tangled up in the DNA, and make DNA available to the transcription machinery. Now let's see how the transcription happens in these higher organisms. First of all, promoter has two essential sequences: a recognition sequence where RNA polymerase binds, and Tata box where the DNA begins to denature. Tata box is basically, as the name implies, is a region of DNA which is rich in the nucleotides T and A, and it is about 25 base pair upstream of the start site where the transcription will start. As I mentioned, eukaryotic polymerase cannot bind the promoter and initiate transcription. It needs help from transcription factors. One of the first proteins that transcription factor that arrives at the location is TF2D, transcription factor 2D. Here I would like to mention that RNA polymerase 2 is the RNA polymerase that is responsible for transcription of structural genes. RNA polymerase 1 in eukaryotes is responsible for making ribosomal RNA, and RNA polymerase 3 is the RNA polymerase which is responsible for making other types of RNAs, SNRPs, we will talk about that later, and also transcription tRNA. That is the function of RNA polymerase 3. But transcription of structural genes is done by RNA polymerase 2. So transcription factor 2D arrives at the location. This transcription factor has also an area, a component which is called Tata binding protein, includes in this TF2D, that binds the Tata box, which is depicted right here. And this is the starting point for the transcription. Once TF2D binds the relevant position, other proteins are engaged. They come in and they start binding the complex, because when TF2D binds the DNA, it changes its own conformation changes, and also it changes the conformation of DNA and makes it available to proteins that could not bind before the change could do so now. So these proteins keep on accumulating, and one of the last proteins is the RNA polymerase 2 in this case, will join this whole complex, the transcription complex, and the transcription will start. So this is how things look like in eukaryotic transcription. We have chromatin remodeling complex. This is sort of, if you want to think about it, it is like a bulldozer. This is a bulldozer in front of the rest of the cellular machinery. The function of this bulldozer is to smoothen the road. It is basically getting rid of the obstacles, which are the histones or the proteins around which the DNA is bound. So it is bulldozing the way, and tagging right behind it is the RNA polymerase, which is right behind the chromatin remodeling complex, and it is doing transcription. Transcription meaning it is producing messenger RNA that can be used to make proteins once it has matured. So additional information regarding transcription in eukaryotes. Many genes also have enhancers and silencers. These regions of DNA could be thousands of nucleotides away from where the transcription starts. It is thought that enhancers or silencers, these regions of DNA, they recruit specific proteins, and these regions, they sort of bend over at the promoter site and it facilitate binding of the transcription factors or the RNA polymerase, and thereby facilitating transcription. So as an additional information regarding the transcription in eukaryotes. As we saw in prokaryotes, their genes are in tandem. We have already talked about what tandem means, side by side, adjacent to each other. In eukaryotes, it's not such. In prokaryotes, we had operons. We had the lac operon, the trip operon, in which the related genes, which had a related function, were grouped together. In eukaryotes, the related genes may not even be on the same chromosome. So when we want to activate genes which play with products of certain genes that play a unified role, they have a unified role in a metabolic pathway perhaps, how do we activate all those genes? The strategy of eukaryotes is different than prokaryotes. Here, for example, the example is if a plant suffers from drought, it results in production of a regulatory protein. A type of transcription factor is produced. This transcription factor can go and bind specific elements in DNA. So wherever a specific region of DNA exists, here we are calling it SRE, the stress response element. When these proteins, regulatory proteins, wherever they find SRE, stress response element, they bind it and they result in activation or recruitment of other protein complexes and result in transcription of adjacent genes. So it doesn't matter if the genes are separated, they are on different chromosomes, they are far apart, as long they have promoter which is recognized by a specific transcription factor, all those genes will be activated once that transcription factor is available or is in active form. So we have seen these two different strategies of how prokaryotes and eukaryotes turn their genes on or off.